Hi, this is Emprah Nagar. Today we are discussing about the cytokine and these roles and cytokines roles and abnormalities and diseases, how cytokines roles in health disorders. So, cytokines are peptide synthesis and it releases white blood cells and tissues macrophages that stimulate or suppress the functional activity on lymphocytes and monocytes and neutrophils and fibroblast cells and endothelial cells. Cytokines these are substances released in leukocytes or other cells control the development of the immune response. Often termed the hormones of the immune system, they modulate the difference differentiation and divisions of hemopoietic stem cells and activations of lymphocyte phagocytes. The corticosteroids were among the earliest compounds found out they have immune suppressor activity. So how means the binding of the glucocorticoids to the receptors blocks the synthesis or releases the lymphokines and cytokines. This results in the inhibition of T cells response and to stimulate the redistributions of lymphocytes from the vascular to the lymphatic system and decrease the number of circulating to T cells and beta cells. The cellular immune response is blunt but also must immune suppressive is seen the humoral response antibody production. Cytokines are soluble proteins that interact with the specific cellular receptors that are involved in the regulations of the growth and activations of immune cells and mediate the normal and pathological and immune response. Cytokines are peptides used by the cells of intracellular communications and for the controlling of inner environment of the cells which are the operate. They are produced by the cells type that have important role in the immune response, inflammation, homeostasis, healing, and systemic response to injury. Many cytokines measured by the bioassay and immune assays. So clinical significance cytokines are inflammatory process. The immuno-inflammatory systems is the complex network the cells of humoral elements that include many cytokines. The typical immuno-inflammatory response is triggered by an antigen. The antigen is presented by the specialized cells that termed as antigen presenting cells, that is APCS, the antigen presenting cells that present an antigen through the either class 1 or 2 transaction. So these are the the transformations of CD4 lymphocytes into the Th1 and Th2 subtypes of currently thought to be the central stage of the immune response. Although unconfirmed, it is thought that the lymphocytes can polarize the Th1 and Th2 cells according to the cytokines combined contaminated with in the microenvironmental in the cells residues. Then CD4 lymphocytes are transformed into the Th1 and Th2 subtype. They produce a characteristic cytokine profiles. So how it is means the regulations of general scheme of development of human CD4 the cells. It summarizes the Th polarized, which is usually occurred due to the an antigenic stimulatory allergens helminthic parasites antigens. So the cytokines and cancer. Cancer are very heterogeneous groups of disease, and the mechanism of malignant transformations and contributions of are very diverse in the different tumors. They are represent disease in which intracellular signal mechanisms have been damaged so as to remove the normal constraints or on cellular growth and replications. In many cases, cytokines forms the part of these controls mechanisms or include the other molecules that perform the functions. For example, some proto-oncogenes and oncogenes code normal, abnormal components or cytokines receptor signal transduction pathways. How means the cytokines may have growth inhibitory properties directly on the cancer cells and cause tumor regression due to the modification of the host tumor relationship or enhance the anti-tumor immune effect. They may also act as a growth factor for malignant cells. As far as clinical laboratory is concerned, the measurements of cytokines in biological fluids may be useful from for monitoring of progressions of some tumors and for therapeutic monitoring when they are used as anti-cancer agents. The regulations of growth and differentiations, the natural interferon, they are naturally assumed great importance in the research for tumor modifying cytokines in view of their growth inhibitory properties on many cells. They can down-regulate the expressions of cellular proto-oncogenes such as MIC, the enzymes associated with the DNA replication, and the receptors for the growth factors such as AGF. In vivo, they have maybe also activate the cytotoxic T cells, and the interleukin 6 is the inhibits the growth of a variety of human cells lines derived from the malignant tumors, breast, ovary, and myeloid cells. Interleukin 6 is a potent growth factor for plasmacytic and myeloma cells. The toxicity for tumor cells. The mechanism of arachidonic acid dependent and maybe involved the production of oxygen free radical that destroys the tumor cell DNA. The tumor necrosis factor can inhibit the tumor cells growth by the directions of cytotoxic effect, inhibits the proliferations of induce the differentiation. The TNF tumor necrosis factor can also stimulate growth of the some of malignant cells. Tumor necrosis factor inhibits the promoter effects. So some of the immune response to the tumor suppressor role of cytokines include the maybe regulatory effects on the immune system. The interferon gamma enhances the expressions of major histocompatible cells. The class 1 and antigens are many differentiations different normal and malignant cell types, making the susceptibility to killing the by cytotoxic T cells. How means interleukin 2 stimulates peripheral blood lymphocytes regarding the subpopulations of them more subtoxic for tumor cells. These subpopulation cells known as lymphokines activated cells imported activated killers killer cells probably forms the natural kappa cells populations. The natural killers popular cells population NK cells, they are responsible for immune surveillance of potential malignant cells. Interleukin 2 is used for therapeutic interventional subcutaneously and the renal cells carcinoma and melanoma melanoma. It is some of the cancer gives the response of significantly better than the chemotherapy. The cells may be stimulated the ex vivo using the lymphocytes deriving plasma peripherals and infused into the patient. The tumor infiltrating lymphocytes may be extracted and stimulated. Significant toxicity limits the dose is interleukin 2. Some of the mediations paranucleoplastics effects of a wide range of neoplast effects seems to be mediated by cytokines. The fever to often accompanies the cancer is mediated interleukin 6, interleukin 1, and tumor necrosis factor. Fevers to typical occur in the lymphoid malignancy these cytokines are released. How the anemia may be mediated by the tumor necrosis factor and thrombocytes and interleukin 6, and cachexia is mediated by the tumor necrosis factor and interferon gamma. Bone reabsorption and hypercalcemia are induced the interleukins 1. So cytokines as a tumor markers interleukin 6 levels are elevated in the significant proportions of patients with myeloma and correlates broadly with the disease activity, proliferation index and survival. Patients with mitochondrial gammopathies and understanding significance generally have normal and low levels of interleukin 6. So how means Hodgkin disease and non-Hodgkin disease lymphoma, there is a correlation between the symptoms of fever and malaise and interleukin 6 levels and elevated concentrations of the soluble interleukin 2 receptors and have been found in the number of malignancies of the lymphoid system and the leukemias. The children with acute lymphoid leukemia raised levels of predict the replace and correlate with the survival. The use of interleukin 2 receptors measurements will have a place in the management of hematological malignancy. The tumor necrosis factor expression or protein production has been determined demonstrated in many cancer cells in lines and biopsies. Serum tumor necrosis factor and serum tumor necrosis factor receptors levels raised in a wide range of malignancies. There is measurements of may be useful for the follow-up the studies. The high proportions of patients with hematological and malignancies have raised levels of CSF and also CSF ovarian like cancers, and the great interest like MCSF together the index therapy and ovarian cancer. So a number of the growth factors such as interleukin growth factor and related derived growth factor and tumor growth factors, and they raised serum concentration various cancers such tumor growth factor alpha and levels raised in the breast cancer and hepatocellular cancer, and urinary some of the T growth factors raised in the head and neck cancers. Finally, we'll come to the clinical significance. The rheumatoid arthritis, it is a sympathetic inflammatory disease, joints and other tissues. That disease initiated generally and predisposed in individual and by activations of helper T cells and responding to some of the other orthogenic and possible of a microbe. So how means the activated CD4 cells produce a number of cytokines that have principles to effects, principles of effects, activations of macrophages and other cells, the joint space on which releases the tissue and destructive enzymes and other factors that perpetuate in inflammations and activations of beta cells and resulting in the productions of antibodies, some of the which directly against self constitutes. The results resultant autoimmune reactions damage and joints and deliver the play an important role of disease programs. The rheumatoid arthritis synovium embarrassingly enriches both lymphocytes and monophagocytes as desired cytokines. So activity like these contains can be major account many features of rheumatoid arthritis and interleukins 1 and tumor necrosis growth factor and cause proliferation and synovial cells and fibroblasts. They will also stimulate the synovial cells and the chondrocytes and secrete the proteolytic and matrix and the degradations of enzymes. So some of these things on interleukin 6 and interleukin 1 and 515 and CSF GM CSF, interleukin 15 and these are the tumor necrosis factor and all the relation about the cytokines and cancer. Thank you.
Cancer.